Hi, I'm Ryan Stevens, and this is an informative speech about the timeline of genetic history. According to Neil Campbell, the first two people that I'll talk about really started the ball rolling on the discoveries that continue to change our lives through genetic science and knowledge. The field of genetics is and has been one of the most controversial and popular areas of scientific study. Beginning in about 1858, Charles Darwin did his study in the Galapagos Islands, comparing traits of species that lived on the islands to the same or similar species with different traits on mainland Ecuador. This led to his theory of natural selection, and in 1859 he published his book, The Origin of the Species, which explains how evolution works. Um, it's natural selection, as in survival of the fittest, something that pretty much anyone with a high school diploma nowadays has some familiarity with. In 1866, a man by the name of Gregor Mendel, who was an Austrian monk, did lots of experiments with pea plants. He crossbred plants of the same species but with different characteristics to see what their offspring would look like. He was perplexed why parents with purple flowers would have children with white flowers. So what he invented, and we still use today, is called a Punnett square. This Punnett square is for two parent plants that have a dominant gene for purple flowers and a recessive gene for white flowers. Now, in order for them to have a white flower, both of these genes would have to be recessive. So these both have purple flowers. Now, if these two purple flower heterozygous trait plants have offspring, out of four offspring, three of them are going to have purple flowers, and one of them will have white flowers. This is something that we still use today to determine what's the likelihood of different traits. The likelihood here is that 75% are going to have purple flowers and 25% will have white flowers. Really just discovering inheritable traits. In 1871, DNA was really identified, according to Arnold, by Friedrich Miescher. He discovered what he called nuclein. Nuclein, which is what we now call nucleic acid, is what DNA is made out of. And the significance of this find wasn't realized for many years. In 1953, the structure of DNA, which we call a double helix, which pretty much everyone is familiar with nowadays, was discovered by Francis Crick and James Watson. They went to visit a friend of theirs, Rosalind Franklin, who was inventing this new microscope, and they actually used her new microscope, and it was so powerful they were able to see the double helix structure of the DNA. Of course, later when they published their findings, they didn't say anything about her. In 1977, the first genetic engineering company, called Genentech, was founded to make medically important drugs by engineering the genetic makeup of bacteria. According to Lane, we still use bacteria through genetic engineering to create insulin for patients with diabetes. In 1983, according to Arnold, Huntington's disease was located in the human genome. That is, that we located where the defect occurs on the actual chromosome, more or less. So by having a general location, scientists were now able to know exactly where to look so that they could find the defect. This is something that still is helping us out today. We're still using the same methods to find a location first, a general location, and then use that location to focus in on and figure out what could be going wrong with genes to fix inherent issues. The Human Genome Project is something that started a couple years later. Its purpose is to identify the location and what each code of the human chromosome is for. Each human has 23 chromosomes, and each chromosome codes for about 20 different proteins. So in each chromosome, there are different, they're called loci or locations, that code for different genetic features, such as hair color and eye color. It's reported to be finished. However, there are many scientists that say that there's no way they could have figured it all out yet. Between 1990s and 2000, there's been many quick advances in the genetic field. According to Lane, in 1990, gene therapy was used for the first time for a 4-year-old girl to try to change the quality of her white blood cells. The method worked, but wasn't successful for very long, and it wasn't really useful until very recently. In 1997, I think everyone knows, we cloned the first mammal, Dolly the sheep. From this experience, we learned that cloned animals keep the age of the gene. Even though the new animal is very young, the genes are old. Dolly died very young because the animal she was cloned from was an old animal. Animals and bacteria continue to be cloned for medical purposes and enzymes, and even pigs for arteries. Now, what comes next? Genetics continues to be controversial and extremely beneficial. Are we playing God? The information gathered in this field has the potential to change our lives in many ways, and to add to many more scientific discoveries. So what do you think? Should we continue?